Hello everyone, Dr. Gribbins here. I have the pleasure of talking about chordates today. So this phylum, Chordata, is one of the phyla of animals, or what we call Animalia. And I am a vertebrate biologist, and so the vertebrates are within this group and an organismal biologist, and this is one of my favorite groups to talk about. So the image here is you got an animal, a chordate here, that has a backbone that are snakes, and I study reptiles and snakes. I study a lot of the non-mammalian vertebrates. Up here is a taxonomy, a phylogeny that you should have seen before. It's the one that's from your textbook, and I'm sure Dr. Milne used it throughout his lectures. And he talked about all these invertebrates. Dr. Milne loves invertebrates. I'm a vertebrate biologist. And so we're going to look at this group here, which looks really small compared to the number of other phyla of animals there are, and it's called Chordata. And in many cases, the chordates are the animals that you're most familiar with, right? So let's take a look at this particular phylogenetic tree. And as you can see in purple, I've added kind of the taxonomic, what they call taxonomic grades onto this. So I've added the taxonomy to it. All right, and as you can see here, the taxonomy in many cases is a little confusing because it doesn't always add up to what the phylogeneticists actually how they classify organisms. Taxonomists and phylogeneticists often have different goals, and that's why it seems so confusing when you look at taxonomists and how they group animals versus phylogeneticists and how they group animals. Taxonomists, they group animals based on characteristics that separate them into usable practical groups. And a lot of times that has nothing to do with common ancestry. Whereas phylogeneticists, they group animals or organisms simply based on their common ancestry. And so a lot of times the results are not the same, or they don't equal each other. And I can give you an example. In the vertebrates, taxonomists often group animals, and I typically teach animals in the tetrapod group, the animals that have 4 legs and walk on land, as reptiles, birds, and mammals. And taxonomists treat them as separate classes, whereas phylogeneticists don't. They actually include the birds with the reptiles. And so there are only 2 classes in that tetrapod lineage. There are the reptilia, which includes reptiles and birds, and then mammalia, which includes mammals. And so there can be differences between them. Both of them are useful. Your textbook tends to give you a phylogenetic setup, but intermingles taxonomic grades in it. And so I've kind of added the taxonomic hierarchy, if you will, to this slide and to subsequent slides on the PowerPoint to hopefully make it a little bit easier to understand the different groups. So if you remember, we started way over here on the left with metazoa, right? So metazoa, we're in kingdom Animalia, and there are a couple subkingdoms within Animalia. There's metazoa, and then eumetazoa. I'm saying metazoans don't have true tissues. Their tissues are in aggregates. They're not as organized, right? And we talked about the peripherents here. The sponges are in that group metazoa. In the subkingdom eumetazoa, eu meaning true tissues, then we have organisms that are diplo and triploblastic, right? And our triploblastic organisms are found in this infra kingdom called Bilateраria. So that infra kingdom, Bilateраria, is broken down into several what we call supraphyla. There's supraphyllum Deuterostomia, and sometimes there's supraphyllum Protostomia. Your textbook has taken the new approach, and Protostomia is actually broken down into 2 supraphyla called Locotrophozoans and Ecdysoans. And you talked about those. Dr. Milne talked about all those particular organisms. We are going to talk about Deuterostomia, all right? So we're going to focus on Deuterostomia in this particular PowerPoint, these next 2 PowerPoints actually. And Deuterostomia is broken down into a couple phyla here, Echinodermata and Chordata, all right? So Chordata is a phylum, and that's where we're headed, right? Where we're going to where we're going to spend most of our time talking about organisms. So I kind of wanted to throw in kind of the hierarchical subdivisions, if you will, of the taxonomy here as we go through our talk. Got a couple images. I try to throw a bunch of images in here. I'll also throw some animations in here after I lecture to kind of spice up the lecture, hopefully. so that you can see some of these organisms moving around, or some of the apparatus I might be talking about that show off maybe characteristics of the organisms that we use to group them either taxonomically or phylogenetically. So we have, an invertebrate here. We got our jellyfish. And we have a vertebrate here, an image I took last year of a cormorant eating fish, right? So a vertebrate eating a vertebrate, if you will. So let's dive into the chordates. So here is a zoomed in version of the chordate group. So we have an ancestral deuterostome down here, right? We're using Echinodermata on the top. You're following my pointer on the top of this phylogenetic tree as our outgroup. And then here are all of our chordates. All right, down here I have some of the tetrapods, some of the what we're going to call vertebrates. Not all chordates are vertebrates. The first two groups here are considered what they call invertebrate chordates, those that don't actually have a vertebral column or a backbone like we do. The rest of the groups, at least your textbook follows, and recent research on common ancestry suggests that all the groups here in purple are vertebrates. So we're going to kind of work our way down and talk about all of these groups, kind of introduce you to them, because this is an introduction to the diversity of animals, right? So we'll start up here with the cephalochordates and the urochordates, and then we'll work our way into the vertebrates. So again, when we start talking about, or you start thinking about studying for this exam, be able to group organisms together, right? So which groups are vertebrates? Which groups are non-vertebrate chordates? Which groups belong to Actinopterygii? Which groups belong to the tetrapods? And so forth, as we start looking at grouping so that you can answer test questions. Again, a lot of these names are Greek-based, right? And so they're like a new language, so you got to work with them a little bit. I try to tell you what each of them mean as we go along, so that you can kind of understand where the name comes from. You can see a number of characteristics that are used here to break up our tree into monophyletic groups. And so we have some ancestral characters that we're going to talk about that make all these animals chordates. So you can see over here in purple, we have the ancestral chordate, right? The ancestral chordate is going to give rise to all of these chordate groups, and therefore has some what we call plesiomorphic characteristics, or ancestral characteristics, that all chordates have sometime during their life cycle. And so we're going to talk about those characteristics now. So what makes up a chordate? How's a chordate differentiated from all the rest of the animals out there? There are 5 main traits, in my opinion, as a vertebrate biologist. I think your textbook talks about 4 of them. I added a 5th one because the 5th one's just as important. And these 5 main traits are, first, a notochord. So all chordates have a notochord. That's where chordate comes from, chord, right? The notochord. So this is a stiff connective tissue rod that runs the entire length of the dorsal side of the organism. The notochord is eventually replaced with vertebrae, but the notochord forms in all chordates. As a matter of fact, for example, in vertebrate development, the notochord develops, and then the vertebrae kind of develop on top of it. The notochord helps with the differentiation of the spinal cord and the central nervous system, and then it is replaced, if you will, by the vertebrae. But the notochord is what developed first, and it basically is a stiff rod of connective tissue that's found in the back of the, or on the dorsal side of the organism. And it's stiff, but bendable. That's what's important, that it actually bends. And the development of the notochord, and I'll show you this on this picture here. This is a picture of Amphioxus. The notochord is this area right here, if you're following my pointer, that runs all the way down and along the organism. All right, and you can see that the animal ends in a tail, right? Another characteristic of a chordate is to have a postanal tail. So that tail now allows movement, right, to push against the water and swim forward. The notochord is what allows the organism to swim forward, because the muscles can pull on it to move it back and forth. And that is the essential importance of the notochord. It allowed these animals to be more mobile as they developed, or as they evolved during the Cambrian explosion. All Urochordates also have a hollow dorsal nerve cord. As Dr. Milne told you, the invertebrates typically have a nerve cord that's ventrally located, and that nerve cord is typically solid. In all chordates, they have at least part of or an entire nerve cord that is hollow and dorsally or on the back. It's located right on top of the notochord or within the vertebrae. Number 3 are what are called pharyngeal gill slits. I hate using the word gill, but that's what your textbook uses. The pharyngeal slits are not necessarily gills, and we associate ourselves with or think about gills in fish. The gills in fish are kind of a different developmental structure that arise from those pharyngeal arches or slits, but are not the actual gills. But these pharyngeal gill slits form in the pharynx. Now, pharynx, that word is throat. So you notice this is the throat region in me, right, or in a human, but we don't have slits, right, as adults. But we did have these pharyngeal slits during development. Matter of fact, the human embryo looks a lot like a fish early in organogenesis, particularly up in the head where cephalization is going on, because the gill slits form and then migrate to form other structures. And some of the structures that these pharyngeal gill slits form, just to, something to give you an idea, is they will migrate to different parts in the head and neck. They form the lower jaw. They form the hyoid apparatus, which in tetrapods at least, which we live on land, which support the tongue. They form the 3 little bones in the ears on either side that are called ossicles that help us hear and air. So these slits become the workbench to form other structures during our evolution. So present in all embryos at least. Number 4 is a postanal tail, which you see on the other side or behind, or what we call caudal to the anus. Even humans have postanal tails during development. So again, these characteristics show up sometime during our life cycle. The 5th one is a thyroid gland. So the thyroid gland sits in our throat and it produces thyroid hormone. Or it is an organ in some of the more primitive groups, some of the more ancestral groups that at least can sequester iodine. Iodine is one of the heavier anions in the body, negatively charged groups in the body, and is concentrated in the thyroid gland. Iodine activates thyroid hormone. So thyroid hormone is very, very important in development, in what we call metamorphosis in vertebrates, and also in what we call metabolic rate. So basal metabolic rate. And all chordates have either a structure that's similar to the thyroid gland or a true thyroid gland. So these are the 5 characteristics that make up a chordate. Every chordate has to have these 5 characteristics. Now, the phylum Chordata has 3 main subphyla. So we have the cephalochordates, the urochordates, and then what are called vertebrates or vertebrata. Or I usually teach it craniata, right? I'm old school. I tend to be very old school, and I like the term craniata, and I'll explain to you why, because of my physiologic background later and my anatomical background. But these are the 3 major groups that molecular data really support very well as far as phylogenetics go. If you are a taxonomist and like to use taxonomy, which I do a lot, use taxonomy, I like to think of this 3rd group as craniata, and I'll explain why in a minute. But the more recent data, there's a really nice paper out there from the Proceedings of the National Academy of Science that was published in late 2019, so really, really recent, that it combines molecular data with morphological data and includes a number of new new fossils. And so they have this, this new kind of rendition of how we should group the organisms. And it's come back to this old school grouping, which is cephalochordata, urochordata, and vertebrata. All right, so those are kind of our 3 groups. And so the cephalochordates are really lack diversity as far as the living representatives of cephalochordates, and include this one species. They're called lanceolets, and I'll show you what they look like. This is an aquatic fish-like organism that's a burrowing organism and is completely marine and lives only in the oceans. Urochordates are what we call tunicates, or we call them sea squirts, is another name we use. And again, they're aquatic they're very non-vertebrate-like. They're sac-like. They look like a ball of jelly, if you will. And they are filter feeders. And then we got the largest group here, the vertebrates or the craniates. And these are chordates with vertebral columns, except for this one group called Myxininae, which are hagfish. And so I'll talk about those. Included a little lizard picture up here. Many of these terrestrial pictures I've taken across the world. I like to photograph. So, but we're going to basically take each of these groups as we go along and talk about them in a little bit of detail. So you got an introduction to kind of the diversity and organization of these groups. So this is what a lancelet looks like inside of an aquarium. All right, so you can collect these guys, you can throw them in an aquarium, and you can take a look at them. They tend to burrow into the sediment. It's to hide themselves, to protect themselves from predation, and they're also simply filter feeders. And this group is called the cephalochordates. And the reason they're called the cephalochordates is because they show this major development, if you will, or specialization to the head. So cephalo means head. If you compare them to other invertebrates, for example, echinoderms, the ancestral deuterostome, or the more what we call primitive deuterostome, right, or basal deuterostome. So this subphylum, which is under phylum Chordata, this is again one of these lancelets, kind of burrows and digs itself down into the sediment with its head poking out, and its head has a filtering apparatus where it can filter water. And that's why they're filter feeders. So again, there are lots of fossil cephalochordates. There are a bunch of them that arose out of the Cambrian explosion 500 million years ago. And then they diversified because they can move and they could hide and they could filter feed. But the only one that is living or what we call extant today is the lancelet. And this is an example of it. And they're pretty rare, to be honest with you. They're what they call locally abundant. If you can find an area where they're at, you can collect these. One area, for example, here in the U.S., there are two kind of areas in the U.S. where they're pretty common. Chesapeake Bay and Tampa Bay, all right, believe it or not. It's a great place to dig through the sediment in Tampa Bay. You can find some of these. You can net some of these and take a look at them. But they're not big. They're about 5 to 7 centimeters. So they're not very, very long, right? A little over 2 inches, 3 inches. They filter food from the water using the pharynx. Again, they use those pharyngeal slits to filter water. The pharynx against the throat. They like to bury themselves for defense. And this particular group is dioecious. So this has to do with how they carry the sex organs. Dio means two houses. So there are separate male and female lancelets. Like us, we're separate male and females. Some of our chordates are monoecious, same house, and have both sex organs in the same individual. But this particular group, the cephalochordates, are dioecious. So this shows you kind of, this is a real image of them sticking their heads out. And if you look here, there's this little oral groove, or what they call the oral hood. I'm circling it with my laser pointer. And what they do is they draw water in the mouth. And then this clear area here in this picture is the pharynx. All right, and the pharynx has slits in it. So if you're looking at this drawing down here, they basically draw water in through the oral hood, and it passes into the pharynx, and it is going to leak out of the slits, entering the body cavity, which we call the atrium, the truculum, the atrium. And then that water leaks out the atriopore down here. So the water comes in the mouth and out the atriopore as it goes through the pharynx. All the food is going to stick to the slits because they're mucus. They have a lot of mucus on them, and they collect particulate matter and algae and zooplankton and whatnot out of the water. And then they pass that into the intestines where they digest it. And then this ends in an anus, right, where they get rid of what they can't digest or use as food. And so they hide themselves and filter feed like this. They can't swim relatively quickly when they move around because of their postanal tail and their notochord. They show all 5 chordate characters as adults. So dorsal nerve cord plus a notochord. There's no background here. We're not a vertebrate. Poorly developed sense organs. You can see the head is developed really as a feeding apparatus. There is an eye spot, and there is a really, really small kind of ganglia that's up in the head region. But really, they don't have a complicated brain. They don't have complicated sense organs. Poorly developed brain and true body cavity. They do have have a true body cavity, or what we call a coelom, and that is the atrium, which is the body cavity that the pharynx sits in and is what collects the water from the pharynx and delivers it back to the environment from the mouth. So that's a little bit about cephalochordates. Let's look at the urochordates or urochordata. It's a beautiful image of one of the more tropical or urochordates or sea tunicates that you find on reefs. They're called tunicates because they have a specialized protein called tunicin that is found in their exoskeleton that makes their exoskeleton kind of strong or hard. And it is unique to the tunicates. So it's only found in sea tunicates, or they're called sea squirts because if you take them out of the water and squeeze them, they will shoot water, much like a squirt gun. So I've played with them before when I've done some snorkeling with manatees. So you can find them mostly in marine environments, but there are some kind of brackish, and some of them even move into freshwater, believe it or not. They're filter feeders. And if you see here, if you look, let's look at this guy right here in the middle. They have basically two openings, right? They're called pores. And one is going to draw water in, and the other one's going to allow water to escape. And what drives water movement in the urochordates and the cephalochordates is cilia. All right, cilia line all of those cavities and beat in unison to force water through the pharynx. So a couple of other pictures of them. You can notice that, for example, if you look at the one here on the bottom right, it doesn't look much like what we would consider a mobile vertebrate, right? And that is because the adult is sessile, all right? Sessile, all right? Sessile means that it sits on the ground, all right? Or it doesn't move. And it's also benthic, which means the adult is on the bottom. All right, so it attaches itself to the bottom and is sessile. It doesn't move as an adult. So most people would, say this is a plant, or it's not alive, or it's definitely not a chordate, right? And a close relative to the vertebrates. Molecular data suggests this group is most closely related to the vertebrates compared to cephalochordates. That's what most of the newest research on phylogenetics using molecular data suggests. But the larva, if you look at the larva, and this is a picture of the larva right here on the left, lower left, is very motile. It's got a long notochord. Look at that long notochord. That's that rod. It's got muscle here that around the notochord that can bend that tail and whip it around. And this guy is pretty microscopic. It's hard to see with the naked eye. You can see it, but it's planktonic. All right, it drifts in the water column close to the surface where the sun is, and it's a filter feeder. So it feeds or filters water through the head of this particular larva. The larva gets to a certain size, and then what it does is it drops to the bottom, and it attaches itself to the bottom and becomes the adult here. And there are a couple siphons, the holes, that actually do the filter feeding. So they have a dorsal nerve cord and a notochord. At least the larva has a notochord, and there is no backbone. All right, it's not a vertebrate. So mesenchyme surrounds most of the organs. So it doesn't have a true coelom, which is weird because this is a coelomate, right? The larva has a better representation of a coelom and less kind of development to the organs. So there's kind of a coelom, but not really. So it's really an acoelous organism as an adult. So it's undergone what we like to, some evolutionary biologists like to call reversal in evolution, back to the benthic state where it will sit on the bottom and filter feed. It's a sac with two siphons. Again, it has a pharynx with slits in it. Water enters one siphon and then exits the other one, all right? And so, for example, in this particular organism here, you can see water would enter at the top. It would come down into the pharynx and leak into the atrium and then come out the siphon on the side, all right? So that is how it works. I have a little video here that shows you, as you'll see, the movement of a dye through both of those siphons. Filter feeders, hermaphroditic, which means they're monoecious. They have both sex organs. Sometimes they release both sperm and eggs. Sometimes they release sperm at one time and eggs at another. It just depends on the species. One interesting fact about these guys is, in Florida at least, they have been implicated in the death of some manatees because they tend to attach themselves to the saltwater grasses or the grass beds that manatees feed in. They filter feed out planktonic organisms, right? One of those planktonic organisms that happened in the last 20 years to become very common when it gets really hot and warm in Florida are the dinoflagellates, right? And then they produce red tides, which have toxins in them. And then manatees, when they're eating the grass, eat these guys sometimes on accident. And over time, they basically build up those toxins in the body, fat tissue, if you will, of the manatees. And often you'll find this large dead manatee and there's nothing wrong with it, right? And when they do an autopsy and they do some toxicology on it, they notice these red tide dyes are really high, which has happened in a few instances with manatee populations. So that's the urochordates. So we've done cephalochordates and urochordates are two invertebrate chordates, right? So sea tunicate or sea squirt are the names, as I mentioned. So now let's talk about the third group. We're going to basically spend the rest of the semester, if you will, talking about this third group, which are the vertebrates, or what we call vertebrata, and I like to call them craniata, and I'll explain why here in a minute. But craniata or vertebrata includes two major taxon, if you were two major taxa. One is called cyclostomata, or the cyclostomes, and the other one's called gnathostomata, which are the gnathostomes. Cyclo means round, stoma means mouth. All right, so they're round mouth organisms. You'll see why in a second. And the gnathostomata means movable jaw, mouth that moves. All right, so that's what the gnathostomes stand for. So we're going to talk about that group next. All right, so here we have a figure out of a recent paper. So again, I mentioned this before. So this is out of a very, very nice journal that's called Proceedings of the National Academy of Science. It's a highly thought of journal. It's peer reviewed. It's really hard to publish in. And what this shows is one of the figures out of the textbook. And this particular group of authors were using the newest molecular data, the morphological data that's used in the past, and then some new fossil data where they use morphology to kind of form a phylogenetic inference, if you will, to the vertebrates. Now, as you already know, the hardest part about soft-bodied organisms is that in which these early groups are, the early group chordates are soft-bodied organisms. It's really hard to get good fossils for them, right? It makes it really hard to do any comparison, at least fossil-wise, of the origin of some of these groups. So they use molecular data. Now you can see there are a couple different hypotheses up here. This is the updated hypothesis, and we're going to use this hypothesis because this is the one your book actually follows. But I wanted to show you the other ones. This is the way that I typically teach the vertebrates. This is the grouping I've liked. I've liked it for a long time. And they're called craniata. So the group, instead of being vertebrata, the third subphylum, it would be craniata. And within this particular group, there would be infraphyla, right? And one of them would be vertebrata. But you see craniata here would be our subphyla. And crania means cranium, right? So the development of the cranium, the skull, and then all of the openings for all of the special organs. And craniata would typically include this group here, which are called hagfish. We call them myxininae. And then lampreys and everything else would be vertebrates. And the reason that they make this separation is based on morphology and physiology. Hagfish tend to be physiologically similar, if you will, to invertebrates. All right. I'm not going to explain to you why, but their physiology mechanisms and their compliance to environmental constraint is very invertebrate-like. Whereas all the rest of what we're going to call vertebrates, lampreys included, and including all fish and tetrapods, are very strict regulators. They really regulate their internal environment much different than the environment that they live in. And so they tend to have more derived complex mechanisms that allow them to do that. And they tend to, at least in that particular way, both anatomically and physiology, make up a much more coherent group we call vertebrata. Over here, we had what was called the cyclostome hypothesis, which was based on the original molecular data that they did on their phylogenetic analysis, showing you basically that we have cyclostomes and we have a group of ostracoderms here based on their morphology. And then we have the rest of what we're going to call fish and tetrapods. So in this instance here, all of the ostracoderms, these ostracoderms, I'll show, you know, are ostracoderms. These are fossil or dead or extinct groups of cyclostomes. So they were kind of grouped together on their own, and the living groups were kind of grouped together by themselves out of simple necessity. We don't have any way to do DNA analysis, right, or protein analysis on extinct groups. We can only do the living groups. And so they would basically put the base of vertebrata with the cyclostomes, right? The ones that we could actually look at, hagfish and lampreys. The ostracoderms, they look a lot more complicated. They look a lot more fish-like. So based on that kind of derived morphology, they were an intermediate group between the living cyclostomes and the rest of gnathostomes. This group looked at a couple of new fossils and suggests that some of the extinct groups should belong, because of the way they look, to the living cyclostomes, which show a lot of basal characteristics, a lot of invertebrate characteristics. The other ostracoderms, the other fossilized organisms, again, still form that intermediate. And then you have gnathostomes here, right? So in essence, what we have here with this new data is kind of moving some of these fossilized forms over onto our basal line here, which has both hagfish and lampreys on it, having a few of the fossilized fish in the middle, what we call ostracoderms, and then the rest of jawed fish, which we call gnathostomes. This is the way your textbook does it. So it's going to break down vertebrata, you notice there's no craniata anymore, vertebrata into cyclostomata and gnathostomata. And that's kind of what we're going to follow. If you take my comparative anatomy class, I will follow the blue. But here I'm going to follow your textbook. Couple pictures. Beautiful hammerhead shark, one of my favorite, what we call chondrichthyan fish or sharks. And then this is a picture of a hagfish sticking its head out of the burrow. You can see its round mouth right on the lower left and why it's called a cyclostome. All right, cyclo, round mouth. So our subphylum vertebrata is going to be broken down into two infraphyla. One is called cyclostomata, and the second one is called gnathostomata. So cyclo means round, stomata means mouth. So these are the round mouth fish, and gnathostomata means jawed one, or jawed form. So they have a lower jaw or a mobile jaw, a hinge for a lower jaw. We're going to do the cyclostomes first. There's two groups. There's Myxininae, which are the hagfish, and then there's Petromyzontida, which are the lampreys. So we're going to talk about those two groups separately. If you look here on the bottom, we just have a phylogenetic representation of this. So we got development of vertebrae. Myxininae, as we'll see, does not have a complete vertebral column, and the vertebrae are just blocks of cartilage, and they're usually only found towards the head, or what we call towards the cephalic region of what would be the vertebral region. And so, you know, a lot of texts would say they don't belong in vertebrates, as I mentioned, but they belong in craniata. We'll talk a little bit about why they're weird physiologically also. But you can see vertebrae are kind of put these organisms in a different category or different monophyletic group than the other groups we've talked about. Petromyzontida are the lampreys. They have a true vertebral column. And then we got our first jawed group of fish down here called the first gnathostome, if you will, which are chondrichthyans. So up here is a picture of lampreys. And these are attached to an aquarium, and you can see the round mouth that comes from cyclostomata, the name. So let's take both groups of cyclostomes. The first one's Myxininae. These are the hagfish. Here are a couple images of hagfish. You can see it's a very eel-like animal. Here are some of the characteristics of our typical hagfish. So these guys are eel-like, and we call them fish even though they're not what we call true fish. They only live, it's the only living organism that doesn't have a skull. All right, so it has kind of a cranium, but it doesn't have all of the sensory structures you're going to see in the other organisms. So for example, it lacks eyes. This is the front of the mouth. Here's the mouth here. and it can open that up. It's got some jaws. It can move out. It is not a real predator. It is more of a scavenger. It's a detritivore organism. It is a mud grubber, if you will. It feeds on dead carcasses, dead organic matter. It is a living fossil. The living forms, the extant forms, look a lot like the ancient forms, the forms that are fossilized from 300 million years ago. They lack scales. They have a very smooth skin that's very slimy, and they're called one of the most disgusting organisms in the ocean because they can produce tremendous amounts of mucus. They can produce copious amounts of mucus if you stimulate them. They wrap themselves in this mucus. It makes them really slimy, really gross, really slippery. It's hard to hold on to them, and that's kind of their only defense mechanism. So I have a video of that associated with this slide. The next group are called Petromyzanthidids, which are the lampreys. And the lampreys are a lot more complex and therefore more derived than hagfish. They actually have a nice cranium. They have sensory structures like eyes, and they smell well. Both Myxininae and the lampreys have one nostril that's on the top of their head for smelling. Lampreys smell really well. That's how they find their prey. They are parasitic, completely parasitic. At least the groups we're going to talk about. There are some free-living forms that actually live in freshwater streams, but the marine variety, which has now inhabits the Great Lakes and works on game fish like this trout here, are wreaking havoc on game fish, particularly small ones, because they can suck these fish dry. And their round mouths are perfect suction cups to grab onto the outside of our major game fish and basically open up a wound that they use to feed on blood. All right, and so here are some of the major characteristics. Again, not a true fish, no scales, smooth skin, produce mucus. They lack jaws. They have a nice round mouth, cyclostome. And we'll zoom in and look at that mouth here in a second. And they don't have paired fins like true fish do. They only have one nostril, not two. And they have completely cartilaginous skeletons. And so what they do is they attach, as you can see here, to the surface of fish. And they do that using the mouth. The mouth is very specialized for this. You can see this is a close-up of the mouth. Look at all the teeth. This is actually called the oral disc. Here are the characteristics here of the mouth. You can see the teeth, so they can suction onto a fish's surface, use the teeth on the oral disc to grip into the flesh and skin and muscle of the fish, and then their tongues have teeth on the ends of them, and they can actually open up a wound on the fish, and the fish bleeds, and then they feed on the blood. Any animal that feeds on blood is called a sanguinivorous. All right, sanguinivorous organisms like the leech feed on blood exclusively. And as I've mentioned before, they've been accidentally introduced into some freshwater habitats where they survive and reproduce and then wreak havoc on the game fish in, for example, the Great Lakes ecosystem. One really interesting fact about lampreys is their saliva has a protein component in it that's is a potent anticoagulant. It prevents blood clotting, which is really important if you're feeding on blood. You don't want a big clot to form in your mouth that's hard to swallow. You want the blood to be thin and to flow nicely. This anticoagulant is so potent and so efficient that the pharmaceutical industry is now in the process of mimicking or synthesizing that protein artificially so that they can produce a better anticoagulant that they can use in the medical field. All right, so those are the cyclostomes. Now we're going to do the gnathostomes. So we got basically chordata, subphylum vertebrata, infraphylum gnathostomata. All right, this infraphyla includes the jawed vertebrates, and there are aquatic and terrestrial vertebrates. The aquatic vertebrates evolved first. Life evolved first in the water and then moved to land. No different with the vertebrates. It's a beautiful fossil showing a fish eating a fish or a vertebrate eating another vertebrate, right? So let's dive into these groups, into the jawed organisms. The first class we're going to talk about in this phylum, infraphylum, is chondrichthyes. So chondra means cartilage, ichthyes means fish. So these are cartilaginous fish. These are fish with completely cartilaginous skeletons. The only place where you have calcium salts that invest or invade the cartilage is in the jaws and the teeth, all right, as we'll see. So this is a black tip reef shark here, a nice representative of chondrichthyans. So the characteristics of chondrichthyans. Again, this is one of my favorite groups of fish, and I'd love to talk about it. I could talk about these guys for days at a time, but the lecture has to be brief because I got to introduce you to some other organisms. So I'll try to stick to the characteristics that we're comparing among different groups of vertebrates. So again, their skeleton is cartilaginous, much like the skeleton of the living hagfish and lampreys. Now, if you remember, before I introduced you to a group of fish called ostracoderms, right? Ostracoderms, their name, ostraco, means bone, and derms means skin. All right, so bone actually arose first in an extinct group of fish called ostracoderms, and it's the skin that produces that bone. All right, and that bone that arose in the ostracoderms will evolve into the scales that we see in the true jawed fish. All right, scales are the derived form of dermal bone that first arose in the ostracoderms. All right, that's what we think phylogenetically or evolutionarily. And so our chondrichthyans have scales in their skin, and these scales are hard and strong and actually pretty sharp, and give a nice sandpaper texture to the skin of the chondrichthyans. They also have paired fins, which is something we haven't seen of yet. So if you look down here at this blue shark on the bottom right, you can see this huge pectoral fin that's sticking off here, right? They're paired or on either side, which helps them turn, maneuver, and prevents them from rolling. They also have paired pelvic fins on the back, right? Or sometimes they're called anal fins, or they have both, depending on the fish. And these paired fins are something that is a derived characteristic of the gnathostomic fish or the jawed fish. They have paired nostrils. So there are two nostrils associated with this. I'll show you a close-up of the nostrils on the next slide. And they have scales. These scales have some bone to them, some calcium salt to them. The teeth are also scales. So they have dermal teeth. These teeth are serially replaced, which simply means they shed their teeth, and they do this often. All right, so sharks shed their teeth. They're the best example of this, right? If you look at the inside of the jaws of a shark, they have several rows of teeth, usually anywhere between 7 and 14 rows. And the base of the jaw, where the skin meets the actual jaw, there is a tooth membrane, and that membrane produces the teeth as part of the skin. And so they can produce tons of teeth. A tooth can be produced in a typical shark about every 7 to 10 days. And so they produce new teeth, and then the teeth replace the ones that are shed. So when a bull shark grabs onto something and a tooth is ripped out, the one underneath it replaces it, and the tooth membrane keeps producing teeth. They produce so many teeth that it is estimated that a mature bull shark will shed 30,000 teeth in its lifetime. All fish, true fish, this includes lampreys and hagfish, have two-chambered hearts. All right, and the chambers of the heart are called the atrium and ventricle. All right, whoops. Atrium and ventricle. And fish have one atrium and one ventricle. As we move into tetrapods, we'll add another atrium, and eventually we'll divide the ventricles into two. So the more basal type of heart is a two-chambered heart. So then fish have that. They're also mostly ectothermic, which means that their body temperature is the same as the environment they live in. Now, there is some great evidence, some recent evidence that there are a few derived shark species like great whites, which are endothermic. They actually maintain a higher body temperature than the water around them, but only by a few degrees. So most of the sharks for us, for our exam purposes, are going to be ectothermic. Okay, some other characteristics. Let's put the rest of the characteristics up here for chondrichthyans. So, as I mentioned, dermal teeth and serial tooth replacements. So this was evolved first in these types of chondrichthyan fish. And there's always this question of which arose first, teeth or scales. Scales probably arose first because the ostracoderms, the bone that first arose in ostracoderms was in their skin. dorsally located, and it evolved into scales most likely. And then teeth evolved, at least in the chondrichthyans, within the mouth from skin derivatives that are very similar to scales. These are the scales on the outside of the skin that look very similar to the teeth that you see in these fish. And the scale name, if you want to know, these are called placoid scales. All right. They're called placoid scales. All right, so placoid scales are found in chondrichthyans, and they are pointed, means pointed scale. And you can see the nice points on the individual scales. Another characteristic that's found in all chondrichthyans and is a what we call a derived character for this clade are what is called a spiral valve. So this picture up here, it's kind of hard to see where my pointer is, represents a intestine that's been opened up. And you can see that the intestine has these spirals of tissue in it that breaks down the intestine into individual or smaller compartments where digestion can happen. So the spiral valve allows for increase in surface area, which makes digestion more efficient. They have excellent olfaction. Olfaction is smell. If you look at this picture of the front, the ventral surface of the nose here, or what we call the rostrum, the front of the shark, you have two paired nostrils. Sharks have an in-current and an ex-current opening to those nostrils, and they can move water very rapidly through them. And they have great olfaction. They can smell a drop of blood in 1 million gallons of water. They also have several other sensory structures within the skin that allow them to sense vibration and to sense electrical current, which is really nice. So the lateral line system is a system of canals or pores on the shark or along the shark's body or a fish's body that have little hairs in them. Those hairs bend when water next to them moves in one direction. And the nervous system can pick up that type of movement of water, which allows them to feel fish swimming around them. This is also what allows fish to school and move in unison with one another using their lateral line system. So it detects movement and vibration and pressure within the water. Sharks have electroreceptors also in the face that are concentrated, especially in the face region on the ventral surface. Again, coming back to this image right here, all of the little dots that you can see with my pointer here, my laser pointer, are the openings to their electroreceptors. And these are called ampullae, is the name given to them. And they're named after a scientist, Lorenzini. Lorenzini is an Italian. I don't speak Italian, so I'm sure I'm saying that wrong. Lorenzini is an Italian biologist who first discovered them. And that's where they come from. Or they're just called ampullae. And this is what the ampullae looks like. What you're seeing on the shark here is the actual pore or the opening. So if you go into that opening, there's going to be a long canal and a swollen bulb on the end. That is the receptor that's called the ampulla, and it has the receptor cells in it. And then these are attached to the dendrites of sensory nerves or to a sensory nerve. And so they pick up electrical current, the movement of electricity through water. And we, or all vertebrates, produce electricity when we move our muscles, when we breathe, when our heart beats, when the brain works. That is all run by electricity. That's what nerves and muscles do. They can, nerves at least can produce electricity. Muscle can react and conduct electricity. These very specialized, highly sensitive electroreceptors can pick up small, minute electrical currents like heartbeat. And so a ray can swim over the top of the sand and feel or hear, if you will, the heartbeat of a fish that's buried in the sand, find it, dig it up, and eat it. Fantastic derived receptors that allow them to interact with their environment. Okay, one more slide here on the chondrichthyans to finish them up. Again, a very complicated, very derived group. Probably the most incredible thing about particularly the sharks is that they haven't changed much over the last 100 million years or so. Or even longer. They kind of came out as almost perfect predators, apex predators, and haven't changed a lot. So if you look at fossil sharks, they look very similar to the modern sharks. Well, the only major difference between them is size. Sharks have shrunk a lot in size. As the teeth showed in the last slide. Let me back up 2 slides. I forgot to mention that. Now that let's go back. So notice here, the tooth difference here. This is a mature great white, the white one, which was about 25 feet. And this is a megalodon tooth. Tooth width and length you can use to estimate size pretty confidently. So if this is a 25-foot shark, this is a 70-foot shark at least. So the big prehistoric sharks were very large, hunted dinosaurs, for example, and during their first radiation where they diverged out and to form many apex predators within the ocean, they would eat dinosaur or reptilian-like dinosaurs that were living in the ocean. And then megalodons differentiated to feed on whale or whale ancestors. And so they were incredible size, incredible teeth. And the reason these teeth are common is because of serial teeth replacement, right? All right, so back to this slide here. Before we do the taxonomy. So here are the rest of the characteristics I kind of want you to know about sharks. This also introduces you kind of the reproductive strategies that vertebrates have also. Sharks show all three reproductive strategies, which shows you, how diverse and derived that they actually are. The first thing that's really neat about sharks is they practice internal fertilization. Most fish dump their sperm and eggs into the water and hope the eggs and sperm meet. Sharks aren't that way. If you look at the male, you'll have their pelvic fins down here that actually have extensions that are called claspers. You can see these elongated extensions down here called claspers. And they stick that into the female cloaca and then deliver sperm through the clasper into the female reproductive tract. So the sperm are delivered directly to the female reproductive tract and then swim to the eggs to fertilize them. Now, females can do three things with their eggs, basically. They can lay them in what are called mermaid purses. And so that's what this is here. This is what is called a mermaid purse. It's supposed to be an R. Mermaid purse, right? That's the name that they've given these. So they have this hardened egg case, looks like a purse, where the baby shark develops inside of that. Sharks that produce egg cases like this, the embryo has a large amount of yolk, much like a chicken has copious amounts of yolk that it develops from. And then the shark develops inside of this egg case, and then the egg case breaks open and the shark is released. So, somewhere around 40 to 45% of sharks are oviparous, right? They lay eggs. So oviparous means to lay eggs. There's no parental care. They lay the egg. And this is because most sharks are pretty small, believe it or not, typically under 3 feet. And most of them are going to live in shallower water or along the continental shelf and can lay an egg case down on a reef or vegetation or something within the ocean that they are close to, right? Because they're close to the surface. Some sharks that are pelagic, that live out in the middle of the ocean, don't want to drop an egg case to the 3,000 feet, right? They actually are going to retain the egg. Many of these sharks do, and they're called ovoviviparous. So they still lay eggs, but the eggs stay in the female reproductive tract until they hatch, and then they release the babies. The ovoviviparous species, some of them practice what's called oophagy. So if you look down here, we have oophagy, right? Oophagy is practiced by ovoviviparous sharks. And what oophagy means, if you look at the word, oo means egg, phagy means to eat. So female sharks will produce unfertilized eggs, extra eggs, if you will, and dump them into the female reproductive tract, and the babies start eating before they're even released. So they're eating eggs in utero before they're released, if you will, from the female reproductive tract. We call that live birth. So they can produce bigger baby sharks. The bigger the baby shark, the less predators it's going to have. Believe it or not, there are also some sharks that are viviparous, like us. They practice viviparity. So they have a placenta, like mammals do. It's a very primitive placenta, or a very simple placenta, but the embryos are actually attached to mom's blood supply and are like parasites. And again, you have live birth. The most derived sharks are viviparous sharks. Well, another really cool thing, some of the viviparous sharks even produce what's called uterine milk. They produce milk in the female reproductive tract that the sharks actually ingest to help them grow really large. So these viviparous sharks produce 1 or 2 pups that are big, all right, that are big sharks that are, sometimes 3 feet long in some of the larger species that are ready to come out as an apex predator, as a 3-foot predator. And so These three reproductive strategies are found throughout vertebrates. Not all of them, not every group, not every taxon practices all three, but sharks happen to practice all three. A couple other things that are on this slide. Most sharks have between 5 and 7 gill slits on the side of their head where their gills are found. They have to keep swimming because they can't move water across those gills when they're sitting still. Most sharks swim all the time, and when they're sleeping, part of their brain sleeps for a while, and then the other half of the brain sleeps for the other half of their sleeping time. Some sharks do have a spiracle, which is on the dorsal side where they can actually move water down into the gill chamber. This is what the spiracle looks like. Water can move in and then go out the gills. Not all sharks have those spiracles anymore, but several of them do. And these are the bottom-dwelling sharks that can actually sit and use the spiracle to pump water over the gills. But your pelagic sharks lack those spiracles and have to keep swimming. Okay, so now this leads us into talking about taxonomy. So now we can talk about kind of the different groups here of chondrichthyan fish. So again, a lot of diversity here. There are two subclasses within the class Chondrichthyes. There are the elasmobranchii and then there are holocephalii. The elasmobranchs include sharks, rays, and skates. Here's a beautiful example, one of the fastest fish in the ocean. This is a mako shark, a greater mako shark. They can move really, really fast to catch prey, up to 50 or 60 miles an hour, they said. Very, very fast. And this is probably one of the weirdest fish in the ocean on my right. This is called a holocephalian or a chimera or a spookfish or an elephant fish. It's called a chimera because it looks like several animals. Some of them look like rabbits and an elephant trunk, and they just are very odd looking fish. These are deep water fish that live in certain parts of the world that live in deep water. And they have a lot of the characteristics of what sharks and rays have. So those are your two major subclasses. So let's take the elasmobranchs first. Sharks, rays, and skates, they lack swim bladders. There's no swim bladder. The swim bladder in fish is used to blow up with air, oxygen basically from the blood supply. That is like the BCD that scuba divers wear. They add air to kind of maintain neutral buoyancy. Sharks don't have those. So they have really large livers if you were ever to open up a shark. And they have a lot of oil that's associated with their body organs. The oil makes them more buoyant. The other major thing that goes along with their lack of swim bladders is they have often what is called a heterocircular tail. This heterocircular tail is basically, as you can see on this whale shark, it's kind of hard to see, has a very tall dorsal tail fin and a very short ventral or belly fin, and that when they move that, it produces lift. So that helps them produce lift. So that heterocircular tail is much different than what you'll see in fish and other bony fish. When we move into the bony fish, I should say, the bony fish, most of them have homocircular tails, which means the lobes are the same size. The elasmobranchs include the largest fish in the world, which are whale sharks. So whale sharks can be quite large, anywhere from 30, 40 feet long, sometimes longer. There are also a couple of other big sharks, like this is a basking shark. Actually, this is a megamouth, which is one of the more recent large sharks that have been found. Both of these sharks are filter feeders. They're benevolent. You can swim with them. You got a scuba diver here. They basically filter krill and small fish out of the water. The largest ocean-going organisms are always filter feeders. A couple more elasmobranchs here. There's a diversity of feeding types. So you have top predators, you have apex predators like the tiger shark. Here's a nice tiger shark down here, right? Highly feared in Hawaii. You have predators and scavengers that eat dead and live things like great whites and bull sharks. And then you have filter feeders like basking sharks, manta rays, whale sharks, megamouths. They actually are filter feeders. So you got a number of sharks feeding on different trophic levels, if you will. Okay, holocephalians, like I said, just a weird group of fish. Here's a couple pictures of these guys. So this is the elephant-snouted one. You can see they have teeth. They have serial teeth replacement, much like sharks. They have heterocircular tails in many. Come on, go back. So they have heterocircular tails in some cases. They have claspers, for example. So they have a lot of chondrichthyan characteristics, but they also have some characteristics that are really odd. So they have an operculum, which covers their gills, and they can move this operculum. It's a bony plate that they can move over their gills. I'll show you a video of when we get to the bony fish that moves water over their gills. And this is very similar to what you see in the bony fish. It's an example of convergent evolution. They also have some weird anatomical conditions, like some of them don't even have a stomach. They just have an intestine. So there's some oddities to the group. So they're called ratfish, rabbitfish, and elephantfish. They live in deep water. There are some populations like in New Zealand that like to eat them. They're supposedly a delicacy. They have a unique kind of sweet taste to their to the muscle, right? To the meat that they have. I've never eaten one, but that's what they say. Most of them eat mollusks and invertebrates on the seafloor, and some of them have a venomous spine. If you're following my pointer here on the top picture, a venomous spine that they can stick up that is hooked to a venom gland, and they can inject venom. And that venom is it usually causes some pain in the predator. The ones that don't have teeth replacement have plates that are actually in the mouth that are pretty similar to what you see in raisin skates. So that's holocephalia. Okay, our last group of fish here are going to be the superclass Osteichthyes within the Anthostomata, and these are called ray-finned fish. And there's a great example of a ray-finned fish, right? This is the lionfish, right? And so you can see the rays that are covered with the skin that make up the fins. And so these rays dorsally can be spines that are like hypodermic needles that are attached to venom glands. And lionfish have bad venom. So do the stonefish, they have bad venom. These guys have been accidentally released into the Caribbean and in the Bahamas, and you can actually find them all the way up through Florida now if you scuba dive. And they're actually actively spearfished as they're trying to control their populations. There's not much here on this side of the world that eats them. They're actually from the Pacific Ocean, the other side of the world. But let's talk about superclass Osteichthyes. So oste means bone, ichthyes means fish. So now these are bony fish. They're going to develop an endoskeleton, the skeleton on the inside, that's not cartilage anymore, but is actually made of bone. All right, so our first class within the superclass Osteichthyes is Actinopterygii. All right, so Actinopterygii, that term, acto, means ray-finned. All right, so ray-finned fish, as I mentioned, are an incredible group of fish. So a large percentage of all fish are in this group, in one particular group within Actinopterygii. And this is the most diverse group of vertebrate, right? That's the first thing you need to know. And your textbook, I think, is a little conservative. They say about 35%. I've seen up to almost 45 to 47% of vertebrate species are fish, and they're these bony fish. So bony fish, ray-finned fish, and Sarcopterygians included, which we'll do next, all have bony endoskeletons. Whoops, sorry. So they all have bony endoskeletons. So their skeleton inside their body is bone like ours. So you can imagine we inherit bone from fish. So they have a gill cover, and I'll show you a video of this, but there's a gill cover. You don't see gill slits. They actually have a bone, a flat bone that sits over their gills. They can move like this in order to ventilate the gills. So a fish that's an Actinopterygian can be sitting perfectly still and able to ventilate its gills with the operculum and then use its swim bladder underneath that to maintain its depth. Incredible energy efficiency by using a swim bladder. A swim bladder is simply a sac they can blow up with oxygen from the blood supply, or they can take that oxygen back up into the blood and remove it from the swim bladder as they get closer to the surface. So they can control the amount of air entering the bladder to maintain neutral buoyancy, the ability to sit in suspended animation, if you will, or in a suspended state within the water column. Almost all of these bony fish, Actinopterygians, have scales. Not all of them. Some of them have very few scales or almost no scales at all, but most of them have scales or some type of modified scale, which again is just the derived version of bony plate that arose in the skin and the ostracoderms. They have different types of scales. We won't go over those, but the scale type also helps us classify the type of actinopterygian we have. All of them are ectothermic. They have paired fins, again, pelvic and pectoral fins, and they have paired nostrils, just like we do. And they can be very diverse in what they look like. They're very niche-specific organisms, or they're what they call niche specialists. And this has to do a lot with their mouth and then their body form, right? So this is a fish, and this is a fish. That looks like a rock, right? It's called a rockfish, and they have venomous spines coming off the rays of their dorsal fin. Some more characteristics here of actinopterygians. So again, two-chambered heart, one atrium, one ventricle, much like sharks and a lot like cyclostomes. They have more efficient kidneys as far as removing nitrogenous wastes. Their gills play the role of the other parts of the kidney, like water conservation and salt removal. But the kidneys here in these fish are very, very good at removing the nitrogenous waste products from protein breakdown. They have a central nervous system. They do have a brain and spinal cord. They have some very nice receptors, specialized receptors like eyes and nose and whatnot that can detect environmental changes, but their brains are much smaller than what you're going to see in the tetrapods. Most of your actinopterygians are oviparous, which means they lay eggs in the environment. And they usually attach them to something, or sometimes they're floating in certain areas. And there is some parental care that you do see, obviously, in some species, but in other species, there is no parental care. They have swim bladders. These bladders sometimes are attached to the digestive tract. They have a duct from the bladder that attaches to the esophagus, or close to the esophagus usually. And so in these fish, they can actually gulp air and swallow it and move it into the air bladders, swim bladders, and they start to act like or look like lungs. And so you're starting to see that some of the special derived structures in fish, like an endoskeleton that's bony, lungs, limbs, all right, their fins, as we'll see in the sarcopterygians, we're going to inherit those things, limbs and lungs and endoskeleton, from fish. Okay, let's look at their taxonomy. So again, we got a tremendous number of species. I know a couple of fish taxonomists, and man, it's unbelievable the number of fish that are out there, and I don't know how they keep them all straight. So we're just going to look at the big taxa. All right, so here are the big taxa. We have a couple subclasses within this class that we're calling Actinopterygii. The first one is Chondrostii, which are cartilaginous fish, believe it or not. And they include a couple species, the sturgeon, which I have up here, a paddlefish, which I'll throw a little video on here of, and then beachers, which are a swamp-like fish that lives in swamps, and they're really elongated. Sometimes you can see them in pet stores, but they're pretty rare fish to find in a pet store. These animals, the paddlefish and the sturgeon, are big fish. Sturgeon is considered one of the largest bony fish in the world. They can be very large, 18, 20 feet long, and they live forever, for a very long time. And when you look at them, they look very shark-like, right? They have heterocircular tails, or tall dorsal lobe to ventral lobe. They have elongated snouts and a mouth that's found underneath or on the belly side. And they have lots of electroreceptors, or receptors, that are in the snout. They've kind of reversed in evolution, if you will, back to what the shark form is. And this allows them to get big and be bottom dwellers. They use these little fleshy appendages here to find invertebrates in the muck and mud of rivers. And the paddlefish are really large fish. They have a long snout or paddle, a rostrum is what they call it, and they are filter feeders. So they've kind of undergone a reversal in evolution. They're considered basal. They're considered bony fish because their ancestors that gave rise to those fish were bony fish. Their molecular genetics also is very similar to the bony fish. So the other subclass is a much larger subclass called Neopterygii. Neo means new fish, or the derived fish. So these are what are called quick-moving fish, or very fast fish. They include two infraclasses, Holistii and Teleostii. All right, Holistii are fish that have slightly ossified skeletons. That is, their skeleton's ossified but part cartilage. And they include a gar and bowfin fish, one species of bowfin fish. The gars can be pretty long and pretty large too. Cartilaginous, having cartilaginous elements allows you to get larger. And they have also very thick scales, the gar do, and sharp teeth. The teleosteae are where most of your diversity is. So remember I said, 96% of fish are actinopterygians, and most of those actinopterygians are right here in the teleosteae. So this is a very diverse group of fish with lots of derived characteristics, particularly to the mouth. The mouth, you can manipulate mouth structure to fit niches, and they can become very specialized, have very specialized niches that they feed in, and that allow them to diversify and fill all of the little niches and environments that you find in the aquatic realm. Okay, so the next class here within the superclass Osteichthyes, so we have Actinopterygiae, and the next class is called Sarcopterygiae. So Sarcopterygiae, your text uses this old school name called Actinistia. Actinistia translates into crossed mast or mast of a boat or cross-webbed membrane. There's a whole bunch of different translations, but it basically literally kind of or figuratively translates into the same thing that Sarcopterygiae means. So sarcopto actually means fleshy or lobe-finned fish. These are also called cross-finned fish or cross-tapered fish. They have pectoral fins and pelvic fins that are fleshy. They're much thicker. And when you go in and you look at a skeleton, these fins actually have the same bones as our arms. There's an ulna, a radius, a humerus. There are carpals and metacarpals that are found in the fins. Then they can move these fins. They can rotate them like you can. Whereas a ray fin fish, when they go up, they can do this. They can raise and lower their fin. That's all they can do. These guys can move them independent of one another. They can move them in a circle. They have some similar joints to some of those bones as we do in our limbs. And so you can imagine it's these fleshy fins that we inherit from an ancestor of the Sarcopterygian fish. So lobe-finned fish include what are called coelacanths. So coelacanths are very rare fish. They're considered a Lazarus species because up till 1938 they were thought to be extinct, and that's when the first one was caught. A second species was found in 1997, which was really recent. These are deep water fish that live in deep water and tend to hang out really close to the bottom in deep water. Sometimes they come up in certain areas of the world to where scuba divers can see them either at night or at dusk or when it's kind of dark. But most of the time they spend in very deep water or on very deep ledges. And often the way that they kind of hang out is with their head down like this towards the bottom. And they maintain that orientation while they rest. And they use their fins like this to maintain that position. A lot of biologists that study these think that's how their hunting position. So when a crab comes up, or a lobster or something that's an invertebrate or a fish that's on the bottom, they can eat it very efficiently. And that's why they maintain these fleshy fins. So this group is an interesting group. Fishermen in some of the poorer places of the world, like Indonesia or off the African coast, try to catch these on long lines. And it's like hitting the lottery because museums want to buy them. They're worth a lot of money. Okay, the last group we'll talk about, the last subclass here within the Osteichthyans are Dipnoia, which are lungfish. So this is a picture of a lungfish. They're a really odd-looking fish. And you can see they have really large mouths, and they're often very elongated or almost eel-like, and they're very slimy. These guys live in temporary water sources and live in water sources that are in areas that are very frequented by drought. And so they live both in Africa and South America and Australia, actually. So there are a couple different places. The Australian group is this group that's best studied. So these guys are air breathers. They are actually what are called obligatory breathers. They have to breathe air. So if you're going to keep them in a tank, you got to leave airspace in the tank. You can't fill it all the way up, or they will drown. It's just very strange to think of a fish drowning. But they're their swim bladders are attached to their esophagus, to their digestive tube. And so these, when you go in and look at the swim bladders in these lungfish, they look very similar to the lungs of an amphibian, like a salamander, for example, or a frog. And so they have very similar architecture to their swim bladders as the lungs of amphibians do. And so they can breathe really well. Matter of fact, if all the water dries out, what they do is they dig down into the mud, and inside of the mud, they blow air bubbles to open up airspace all the way to the surface. And then they breathe air in a hibernative state for long periods of time, up to 2 years, until the water or rains come back. They have very slow metabolisms and can basically store enough food during the time that they're sitting in the pond to accommodate for these lean times during drought. So they have gills and lungs, which is very interesting. The South American group typically don't dry out very often. They just live in very swampy, very shallow water that's very warm. And again, as I mentioned, warm water doesn't hold oxygen very well. So it's much more efficient to live in those warm water areas by breathing air or gulping air. All right, so as you can see from the bony fish, particularly from Sarcopterygians, we are going to, as a tetrapod, inherit through our evolution the endoskeleton, the fleshy fins, and the lungs, right, from fish. So fish actually evolved these structures to be better fish, but that gives you then the basic model you need in order to move on to land, for evolution to work on those characteristics of those derived fish to allow them to, during their evolution, move on to land. So tetrapods are nested in the Sarcopterygian line, kind of, as far as, or in the lungfish line, however you want to look at it, as far as where they get a lot of their structures from bony fish. Okay, so that's the end of my lecture. I'll put another lecture on. We'll put another lecture up here soon so that we can finish up chapter 34, which we'll cover there for our last exam. Have a great day.